everyone. So I'm going to do or demonstrate at least parts 3 and 4 of the lab. So for those of you that came in on Monday, we did parts 1 and 2. So I'm just going to very quickly recap what we did, and then I'll go through and demonstrate some of the key points for parts 3 and 4. And then by tomorrow, we should have your final results. So tomorrow, I'm going to zoom in again at some point to show you your final results. So it's usually when we do this in class, it is definitely a 3-day lab because there's long waiting periods between the different points, between the different parts. So let me just switch the video here. I'll switch it here. There we go. Okay, so can you guys still hear me okay? Yeah, perfect. Okay, so just to recap what we did, last day we did parts 1 and 2, and the point there was to first get some DNA, get a sample that we could use to get some DNA. So for that, we used our cheek cells. So this is just a picture just to remind you of what cheek cells look like. We would have looked at this in one of the labs earlier in the semester, and I think last semester we looked at them as well. So cheek epithelial cells, you can see there's a nucleus in each one, and that's where the DNA is. And they're easy to collect because they're very loose on your cheeks and easy to pull off. So we did a procedure where we rinsed our mouths with the saline solution, swished around, and then spit that into a cup, and then took a little sample and concentrated it so that we'd have something to work with. Then what we needed to do was get the DNA out of the cells and kind of get rid of all the other stuff that we didn't want, because there's a lot of other stuff in those cells too. So we did a boiling procedure using Chilex. The Chilex is there in part to absorb some of the other stuff from the cell that would get in the way of the PCR procedure. We did that. We then did a few other things, transferred our clean DNA sample into a tube and then stored it for a little bit. Then we made a primer and loading dye mix, which also had a, we had a little bead. So between the different things we added there, it was all the stuff you needed for PCR. So it was the polymerase, which is the enzyme that actually works in the PCR process to extend the DNA and basically duplicate it, and then all the other kind of cofactors that are required. So we added all that stuff in, then added a tiny little piece of our DNA in there, and then put it in the thermocycler and amplified it. We did 35 cycles. So a cycle is basically one set of these procedures. So there's a denaturing step that opens the DNA up. Annealing is where the enzyme sort of attaches to the DNA, and extending is where it sort of does the photocopying. So that's one cycle. We did 35 of those. And as I talked about in class, for those of you that were here, it gives you the option of doing either 30 cycles or 35 cycles. 30 cycles is if you're using ethidium bromide, which is a much more sensitive dye. But we don't use that because it works really well, but it's also very toxic. So anything that can attach to your DNA has the chance of being very toxic, and ethidium bromide is very toxic to people, and that's why we don't use it, because you have to be very careful if you use it. So instead we're using methylene blue. So I had to do 35 cycles because methylene blue is less sensitive, so we needed a bigger final sample in order for something to show up at the end of this whole thing. So the PCR machine went through this cycle 35 times. And then I put those little samples into the freezer overnight. And then I've taken them out and now gone through steps 3 and 4. Okay. So steps 3 and 4. Step 3. So now we have, after step 2, we have this. In theory, we should have a whole bunch of copies of our DNA, enough that we could actually see it when we run the gel later on. But what we need to do is cut it up into these fragments that will allow us to detect this bitter tasting gene. So if we didn't do this step 3, then you would just have hunks of DNA and it wouldn't really tell you anything. So we had to add this special enzyme to it to get it to digest. So I did that earlier today, and then I put the samples into the thermocycler again at 37 degrees to incubate and 37 degrees, that enzyme really likes that temperature, so it works really well at that temperature. So we did that for half an hour to let it do its work, and chop that DNA up into little fragments. Now, before I did that, I also saved a sample of the undigested DNA so that we can compare on the gels between the digested and undigested. I'm going to show you just a demonstration of how I did this in a second. And then the final step will be to put these samples into the gel and run electrophoresis to separate out the different bands. Okay, so what I'm going to do here now is show you quickly how I did step 3. And then I'm going to take a little pause and get set up to show you the gels. Okay, so any questions about that whole procedure there? No? Okay, just stop me at any time if you have questions. Okay. So before I show you this, so in this DNA work, it's good to keep samples confidential. So while we were in the lab, we did have people put their initials on it, but now I've kind of made a color-coded legend so we don't know whose sample is which. I do know it's underneath this sheet, but just so you guys don't know which sample is which, and I will give this to Maria so she knows. So I've made up 2 fictional people to do as an example, so Seahorse and Zelda. Those are our fictional characters that I will do part 3 with. And I just wanted to mention this. In all my time working in a lab, I've always found color coding to be an extremely useful technique because it cuts through all the thinking and it's just automatic. So it makes it a lot easier to not mix up your samples if you can use colors. So that's why I've done it this way. Okay. So here is Seahorse and Zelda. Here's the sample that's gone through the PCR machine, 35 cycles. So in there, if we could see it, we would see billions of copies of the DNA. And this is the same process that's being used for all these COVID samples. When you hear the PCR technique, this is exactly what they're doing. They're putting a little fragment of the virus DNA into a PCR and running it. I think they're doing it 30 times now to make all these copies so that they can see if the virus is there or not. So I have that. It also has this pink solution in it, and that is the loading dye. So that is there so that when we load it into the gel, the sample will sink down into the bottom of the little well and then travel through the gel properly. Okay. This is my little tube for the undigested. So what I'm going to do here is transfer 10 microliters of this into the undigested one, and then I'll have about 15 microliters left to digest. Okay, so 10 microliters. Okay, so again, I have the pipetter set to 10. I'm going to take the sample and mix it up a little bit because probably the DNA may have settled down a little bit, so you want a well-mixed sample. And then I'm going to suck up the 10 microliters and then just put it over here. Okay. There we go. So there's our little sample that we've saved that we're not going to digest. So we're not adding the enzyme there, we're just adding it into here. Okay. Now I have my special enzyme, this HEY3 it's called. Just to give you an idea of, again, this is relevant for all this COVID testing happening. At the beginning, when they were doing the testing, there was a lot of delays. And part of that is because these types of enzymes and reagents are very expensive and hard to produce. So this little vial costs about $100. And you can see that's only about like maybe 100 microliters of solution. But it's because it's hard for them to produce a lot of it. So that was part of the delays when they were first starting the COVID testing. Okay, now I'm going to be adding 1 microliter of this into here so that we can get it to start doing its work in digesting the DNA. Okay, so I have my pipetter set to 1. Again, this is the appropriate pipetter. Got the little tip on it. Okay, I'm going to take 1 microliter. You can actually see it in there. It's a tiny amount, but I can see that it's in there, which is good. And then I'm adding it into my sample just on the side so that I know it went in. Okay, so you can just kind of see it there. You see that little dot? And I like to do this when it's very small volumes because otherwise it's hard to know if it actually went in there or not. And then the next thing we would do with this is centrifuge it. Okay, so that's just to get the enzyme down to the bottom. So those of you that were here would have seen these devices. This is a centrifuge. If you're working in labs later on, you'll use these a lot. They just spin things really, really quickly, and it's a good way to just separate things or like concentrate things down in the bottom of the tube. It's often used for things like, I used to use it to separate out algae samples. So we would centrifuge to get all the algae down to the bottom, and then we'd have a super concentrated sample. And we used it on Monday for our cheek cells as well to get them down so it's really concentrated. Okay, so that's what we would do. And then that goes into the PCR machine for 30 minutes at 37 degrees, so that little bit of enzyme that I just threw in there can chop up the pieces of DNA into the appropriate little fragments. So I'm going to show you guys the PCR machine because some of you wouldn't have seen it if you weren't here last day, just so you can see what's going on over there. So I'm just going to switch the camera for a sec. Can you guys hear me? Yep, okay.
Yes.
Okay, good. There's a microphone on this webcam. I just want to make sure it's working because I'm way across the room now. So this is our great PCR machine. It's basically, it just is able to heat and cool things really quickly and program for all sorts of different cycles. So on this little computer unit here, I was able to program it for the appropriate steps of the appropriate cycle. And for this particular process right now, I programmed it to 37 degrees and then put the samples in and let them digest. So we actually have So those are the samples from Monday. Okay, so they've been digesting for half an hour and they're ready for the gel. So I'm actually just going to leave those in there for now. I'm going to load them up after actually because I need to take a bit more time with it, but I'm just going to demonstrate using the seahorse and Zelda samples for you guys. All right. Switching the camera again. Now, is there a question? Can't hear you. Okay, sorry guys. Okay, so you could not hear me over there with my beautiful monologue? Okay. Sorry.
Just a little bit. You sound very far.
Okay.
We missed a bit, but we can review the thermal cycler again. We'll review it together again.
The Zoom might have been switching the microphone back and forth, so sorry about that.
Yeah, no worries.
Sort of, yeah. Okay, so now what I'm going to do is, if you don't mind, I'm going to pause the video for just about 5 minutes so I can get the gel set up, and then I'll show you the gel loading, and we'll start the gel up just so you can see how that looks.
Okay, sounds good.
Okay, I'll just pause you. I mean, stop my video.
5 minutes, yeah?
Okay, so before I get to this, so here I have the gel that I've created. What this is, these trays work as both electrophoresis to run the electrophoresis and also as a casting tray for the gel mold. So I poured in some agarose, which is the media we're going to use. It's 2% agarose. So it's just like if you're making Jell-O at home. It starts out liquid if you heat it up. And then when you pour it and let it cools into more of a jelly texture. Okay, so now it's cooled enough so that I'm ready to set it up. So I have this comb in here that is the template for the little wells where the DNA will go. To take that out, I'm going to squirt it with a bit of water and then pull straight up. There we go. And now you can just sort of see there's some little pockets in there. That's where our samples will go. Then I'm going to take this out. So you can see these gaskets here. So those were to help keep it all in place. Do that. And now I'm going to set it up. So we need it to run from negative to positive. So I'm going to put it like this. This is the negative end. This is the positive. Okay. Then I'm turning it like this so that it corresponds with the samples we're going to be loading. And now I have to pour some buffer in there. So when we're running this, we need the gel, which is the matrix that the samples run through. And then we also put a buffer on top of it. So liquid buffer. So I'll pour that into the tray. See it. This one's called a TBE buffer. There's a whole bunch of different types of buffers depending on what kind of work you're doing. There we go. All right. Now it's time to load things up. So I've made a little legend about what I'm loading where. So here's the legend. And you can see we have a ladder, which is like our kind of standard. That will have some fragments of known sizes to help us calibrate everything else. And then I'll have my digested and undigested samples. Okay, so each person gets 2 rows. Okay, so first, and I've also written the volumes down here just to make it easy for myself. So I'm going to start by loading 20 microliters of the ladder. Take this back off. And when you guys do this, if you are here doing it, you usually get your tray into place because wherever you're going to run the gel, because you don't want to move it once you've loaded the samples, just to avoid disturbing them. Okay. I'm just adjusting my pipetter. Okay, so 20 microliters. This is the ladder. It's also got a loading die. The blue is a loading die. So that's so that we actually can see that it's gone in, and it also is a bit heavier than the buffer, so it will sink to the bottom of the well. Get a new pipette tip. And I'm gonna mix it up a little bit just to make sure it's well mixed. There we go. Then I'm going to load it into there we go. Okay, so that went smoothly. Now I'm going to load up our seahorse sample. We need 10 microliters, and I'm loading up the digested one first. Oh no, hold on for a second. I just have to check something. I think yes, I think I mixed up the order a bit, but that's okay. So it's actually supposed to be 16 microliters of the digested and 10 microliters of the undigested. I'll just change that. Okay. Get a new tip. Now this part is always a bit nerve-wracking because that's all the sample we have. There's only 16 microliters in here and we need all of it. So if anything goes wrong, then that's it. It's gone. But that's okay. I'm sure this will work. And it's just kind of a pseudo sample anyway in this case. Okay. There we go. Okay, so now I'll just do one more and then I will run it so you can see what happens. And I of course, you're not seeing this, but I'm getting a new tip in between each sample because we want each sample to be clean and uncontaminated. There. All right. So samples are all ready to go. Now I'm going to hook this up to a high voltage machine and run some electricity through it. Okay. be a bit shiny, but I'll turn some of the lights off so you might be able to see it. So here's our high voltage machine. Sorry. So you can put up to 3 gels on each one of these. And I'm just going to hook it up. And then, like I said, I'll turn it on and we'll run some power through this thing. Are there any questions while I'm getting these wires all set up?
No, I just wanted to mention though about the buffer. So I was looking up the buffers. So they are to keep the pH steady, but also they have lots and lots of ions. So that electricity will run well through the buffer. Yeah, distilled water doesn't have any ions, or not very many ions anyway. So the buffer creates that environment for the charge, the current.
Robyn, I had a question about the wells. Like, so it's kind of hard to see, but you put it into the hole that was like the well, right? So there wasn't really any gel there. Okay.
Yeah, exactly. That little comb, where did I put the comb? A little comb that I had in there that I took out at the beginning. Oh, this thing here. That makes these little dents in the gel. So it kind of makes little pockets in there, and that's where I can load the sample. So if you imagine making some Jello and just sticking like a little mold in there, then when the Jello hardens, you pull it out and you have like a little pocket.
Thank you.
And the pocket is in the gel, so you have to put your sample into the gel itself. Yeah.
All right. Plug this in. Okay, so this part I just need to make sure everything's set up properly because I'm dealing with some electricity here. It looks all good. And I'll press run. Okay, and now it'll just take a sec. I'm just going to turn the lights off so we'll see if we can, that'll help us to see things a bit better. Okay.
How long does this usually take?
So usually we'll run this for at least 30 minutes, and then I just check how far the little bands have moved. And if they haven't moved far enough, I'll give it another 15 minutes. So I think it says, I have to double check. I think it says they have to move at least 10 centimeters from the origin. So the origin is where the wells are, and that's to allow the adequate separation. So then, yeah, sometimes I do have to let it go a little longer. And so it's hard for you guys to see this, but if you were here, you would see there's a bunch of bubbles in the buffer now, a bunch of tiny little bubbles that tells me it's working. And we would we'll start to see these little bands moving. Okay, it's not going to be that dramatic, so you won't see too much here, but basically this allows you to basically know that it's working, right? So that band represents kind of the front of the little DNA sample. And at some point along the way, the sample will sort of drop out into the agarose depending on its size, but then the dye will keep moving. So depending on the size, some fragments move further if they're smaller, and the bigger ones stay behind, drop out sooner. Okay. And then, so after this is done, after the 30 minutes up to an hour possibly, I would take this whole gel out and then I put it in the methylene blue stain, and that will stain the DNA. So if you remember, you use methylene blue for your cheek cells because it stained the nucleus blue because the nucleus has a lot of DNA in it. It does the exact same thing here. So it will stain the little chunks of DNA. And then you have to de-stain it as well because at first the whole gel is so dark that you can't see the bands. So then I de-stain it in plain water, usually for a couple hours or overnight, and then the little bands really stand out nicely. if we were doing this with ethidium bromide, we would instead use UV light. So you wouldn't see them, but when you put it under UV light, they glow, the bands glow. Okay. Yeah, there we go. So you can see how this one here is starting to get wider. So that means that we're seeing that it's moving. And I can see from here, they've moved, the origin is here. So they've already moved like a centimeter or so. So that's it. I'm not going to make you sit here and watch this for half an hour unless you want to fall asleep. But
It's actually kind of cool. You can see it moving. That's awesome.
yeah, it's like one of those relaxing Netflix like barge videos where you just watch a barge going down a river for like 4 hours or whatever. So yeah.
And
So,
what happens now, Robyn?
So I let these move. I let these run for at least half an hour, and then I check how far they've moved. If they haven't moved far enough, then I give it a little bit more time. So I'll start it up again.
Okay.
So right now I'm now going to load your samples up. I'll do that behind the scenes and come back tomorrow and show you the results.
Okay.
Just because I want to be careful with your samples. I was worried if I demonstrated in front of you and I messed one up, then you would never forgive me. And also, yeah, just so I can carefully load them and, but yeah.
Yeah. Okay. Yeah,
Yeah.
it's moved quite a lot now.
Yeah, it's actually moving pretty quickly.
Quite wide. Yeah.
Yeah. So I have it
Cool.
at, I think it's, how many volts? I have it set to 130 volts of electricity going through here.
So
That-
if the DNA or its RNA from the virus were amplified and put into the gel, then you would know it was a viral RNA because it traveled a particular distance.
Yes. Right. Because
Yeah. And so for our ladder that we've got there, that ladder is made up of different lengths of DNA that are known. So whoever made up the sample of the ladder knows exactly the size in base pairs of all of the DNA pieces that are in there. So when they travel, you can measure how far they've traveled and plot that against their size in base pairs. And then you can get an idea of the rate of travel.
I don't know if it's somewhat relevant, but it reminds me of a procedure that we did in our chemistry lab, thin layer chromatography,
yes.
where you just know you have a standard compound, and you know the length that compound will travel on a silica plate, and then you put an unknown compound on the same plate, and you just see how far it travels. And if it travels the same distance, then you know that in the unknown compound, you have that molecule or whatever it is that you're looking for.
Yeah, that's that caffeine lab, right? Yeah.
Yeah, that's right.
Yeah. Yep. Yeah, they're sort of similar. I mean, it's slightly different, but yeah, it's similar idea. Exactly.
Yeah.
Yeah. I mean,
Is the
totally.
same. Yeah.
Totally.
Yeah. That's a great comparison.
Yeah.
I'm gonna put that where the origin is. You can see how far it's traveled. So that's about where the origin is. Yeah. Great. Okay.
Wow. Cool.
So that's my demo. So
Nice. Well done.
yeah, if you have any other questions, you can let me know. Otherwise, I will log off and then go to work, carefully loading everyone's samples up and doing this to them.
Okay. Thanks.
Thank you,
Yeah.
Robyn.
No problem.